Well, hi everybody, and welcome to our first CellMolec review video. This is the week 2 review video. So super excited to formally get this class underway. Kudos to all of you for getting your videos watched and for starting the semester strong. And hopefully we'll get into a pretty decent rhythm this week, and this is the rhythm that we'll keep to for the rest of the semester pretty much. So we'll try to keep this as brief as possible, but we also want to hit on all those topics that the majority of you reported were confusing on your video watching and whisks. These are the 3 topics that really bubbled up to the surface so far as terms of being confusing to the majority of you. From the first lecture, it's the Avery Experiment. Really everything else seemed like it was very much review for all of you from genetics, general biology, and even from some high school classes. And then move on to some of the more technical aspects of DNA, specifically chromatin regulation. We'll talk about histone deacetylation complexes, or HDACs, histone acetylation transferases, or HATs, and chromatin remodelers. And then we'll end by recapping the Histone Code. I think that just went by a little too quickly in the video, and we'll give it its due time and attention. So, I guess without further delay, let's talk about the Avery experiment. So when Oswald Avery was doing his research and having his lab, a really burning question at the time was, what is the mechanism for containing and transmitting information in living cells? We have to think back to the naivety of the time, where we really didn't have a very solid understanding as to how cells contained or transmitted information. We knew that cells had physiology. We knew somewhat about enzymes and proteins. Obviously, proteins were critically important to life. We also knew that information was being transmitted because the field of heredity and genetics had been around for a long, long time. I mean, going all the way back to Mendel, obviously we knew that baby pea plants would have some direct connection to the parent pea plants, and we could see traits are genetically transferred. So there were all these pieces of the puzzle kind of floating around in early science and early biology, but there wasn't a clear understanding as to what biomolecule was doing what. So Avery basically did 2 rounds of experiments that conclusively demonstrated what the information-carrying molecule must be in living cells. We know now that that's got to be DNA, but think back to Avery's time. DNA was seen as far too simple, far too regular, far too repetitive to be containing any complex information. So DNA was largely put off as not being the primary molecule to do this. People were pretty sure it was proteins that were containing information, but they knew proteins were short-lived, and so there was a paradox there. So that sets the stage for Avery's experiment. His burning question is, what contains and transmits information in biological systems in living cells? So with that context, hopefully Avery's experiment becomes a little bit more clear to follow. Let's go back to those ground rules. He had 2 types of cells, avirulent cells, avirulent, which basically means harmless or not disease-causing. These cells appeared rough under a microscope, so they were called an R strain or an R-type cell because of that rough appearance, R for rough. And because these were avirulent and harmless, these did not kill mouse cells. So mice lived. Mice could be infected by these R cells. These R cells could take over a mouse and propagate in them, but they didn't cause symptoms. It was an asymptomatic infection, so avirulent, can't cause harm. And then we have the virulent cells. Now these are the harmful cells. They appeared smooth under the microscope, so they were given this S designation, but these, because they were virulent, these killed mice upon infection. So very symptomatic. Symptom of S cell infection was death. To be clear, the R and the S cells are the same species of bacteria, and we already know this in our common everyday occurrence. I'm sure you've heard of E. coli infections. You see it on the news sometimes. Romaine lettuce has been contaminated with E. coli. It's making people very sick. Uncooked food products are contaminated with E. coli, making people very sick. So E. coli obviously is virulent. E. coli makes us sick. But also I'm sure you learned either in high school or in general biology, maybe even in microbiology if you've taken it already, that our gut is coated with E. coli. We've got more E. coli cells inside of us right now than we have our own cells. Human body is about 1 trillion human cells, and you've got somewhere in the neighborhood of 10 trillion E. coli cells in you right now. And for most of you, if not all of you, you're not sick. So how can E. coli infection be making people sick when they eat contaminated romaine lettuce, yet we've got all this E. coli in our gut and we're not sick right now? It's because just like Avery's cells, there are virulent and avirulent strains of E. coli, and we house the avirulent strains normally. But if we get some of those virulent strains in our diet, then they can make us sick. So same thing going on here with Avery's cells. 2 different kinds of bacterial cells, one that can kill a mouse and one that can't. So Avery did a bunch of experiments. He would take his nice, cute little mouse, chubby mouse I have here. Let me make it a little bit cuter. and he would inject in that mouse, our cells. And this mouse remained cute and happy because with an R cell infection, that's the avirulent strain, the mice are not going to die. He repeated that experiment and injected, pretty sad and gruesome, right? And this is not so cute of a mouse. He injected in a separate set of mice S cells, and the S cells propagated in that mouse and killed it because the S cells are virulent and deadly to a mouse. No big surprise. They knew this already. R cells can be injected into a mouse and the mouse lives asymptomatically. S cells are injected into a mouse and the mouse dies. Then what he did was he took S cells, which are virulent and can kill a mouse, and he boiled them. Boiling, of course, kills any living thing. Kills a lobster, kills bacteria, kills these S cells. He boiled them, and then he took the boiled S cells that were known to be inactivated and killed, and he injected those into a mouse, and the mouse itself, lived. So what Avery proved was already known for a long time, that killed, heat-treated, boiled virulent cells could not cause infection. Boil those S cells and the mouse is going to live because the S cells have been killed by the boiling. And it should be mentioned, he also boiled R cells just to see what would happen, but the R cells, when they were boiled, didn't kill the mouse because the R cells didn't kill the mouse when they were alive. Then he took S cells, he boiled them. We've already proven that when boiled, these are avirulent. These can't kill a mouse when they're boiled. But after boiling the S cells, he mixed them with R cells. So what you have here is a composition of living R cells and heat-treated, boiled, killed S cells. And Avery already demonstrated that R cells, when alone, can't kill a mouse, and S cells, when boiled, can't kill a mouse. So what did you think Avery's hypothesis was here? His hypothesis was that these mice should live if injected with this mix, because if R cells can't kill a mouse and boiled S cells can't kill a mouse, why in the world, when you just mix those two safe things, would a mouse die? So he did that experiment. He loaded those heat-boiled S cells with living R cells. He loaded them into a syringe, and he injected that into a mouse, and lo and behold, the mouse died. Why? Well, what Avery quickly realized was that information was somehow being transferred. What information? The information for virulence. Avery realized that the information for how to kill a mouse persisted beyond the death of the S cells. In other words, whatever the S cells contained that gave them the ability to be virulent, whatever information was in the S cells to allow them to be virulent, remained even after those S cells were boiled. And not only did it remain, but it was transmittable. We know these S cells were killed by the boiling. The only living things in that syringe were R cells. So somehow information had been transferred from the S cells to the R cells so that this mouse could be killed. In other words, the residual information from these dead S cells taught the R cells how to be virulent. So Avery knew he was on the right path. Somewhere in that boiled S cell mix were the molecules that contained the information for virulence. And remember how we started this mini review? That was Avery's question. What contains the information in living cells? So again, Avery knew he was on the right track. So what Avery then did, because biochemistry was in its infancy at the time, but there was some biochemistry there, was he started all over. He took those S cells, he boiled them. Now he already knew boiled S cells could not kill a mouse, but he already also knew that the information for virulence persisted in this boiled sample because he had just seen that information teach R cells how to kill. So he knew the information containing molecules was still present and still carried that information despite the boiling. So after boiling and inactivating those S cells, he fractionated them. For those of you that haven't had biochemistry yet, fractionation just means separation. You can size fractionate, which means you separate molecules based on size. If you run a gel in any class, that's what a gel electrophoresis is. It's size fractionation. You separate DNA based on size. You can do charge fractionation, chemical fractionation, ionic fractionation. You can do all these different types of fractionation. Just means you separate things. So he separated this boiled mix, and he separated it into the known biomolecules. I don't know why I have 3 there. He separated it into nucleic acids, which is RNA and DNA, and then separately from that, he purified all of the proteins. Oh, that's why I had 4. And separately from that, he purified all of the sugars, and separately from that, he purified all of the lipids. So he fractionated this boiled S cell mix into the 4 major categories of biomolecules: RNA and DNA, protein, sugar, lipids. Now each of these were distinct and separate. And then what he did was he took R cells, which he already knew could be taught. He already knew that somehow these R cells could take up information and become virulent. That's what the initial experiment proved. And he mixed with these living R cells individually each of these fractionated components. He took the lipids, and only the lipids from the dead boiled S cells, and he mixed them with R cells, and he injected that into mice, and the mice lived. So that meant the lipids could not contain information. In other words, the lipids couldn't teach R cells how to kill. The information for virulence was not contained in the lipids. He took the sugars, and only the sugars from the boiled S cells, and he mixed them with the R cells, and he took that and injected it into mice, and the mice lived. If the information for virulence was contained in the sugars, these sugars should have been able to teach this R cell how to kill a mouse, and it didn't. Sugars did not contain the information for virulence. He took the proteins from that boiled S cell mix, and he mixed them with R cells, and he injected that into the mouse, and the mice still lived. Now, you and I know that it was, in fact, the proteins of the R cell that was conveying virulence. But when Avery boiled the S cells, he also denatured and inactivated those proteins. So those proteins were cooked. And we also know now that proteins do not contain information. Proteins are the manifestation of our biological information. So these mice still lived. But when he took the RNA and the DNA, and specifically the DNA, from those R cells, from those boiled S cells, when he took the DNA from the boiled S cells, and only the DNA, and he mixed that DNA with R cells, and then he took that and injected it into mice, then and only then did the mice die. Interestingly, if Avery took only the DNA and injected it into mice, the mice lived. DNA in and of itself is harmless, no matter what information it contains. So it wasn't the DNA that was killing the mice, it was the DNA that was teaching the R cells how to kill the mice. In other words, the information for virulence, the instructions for being a virulent bacterial cell were contained in the DNA. And that DNA could be taken up by avirulent R cells. And when that DNA was taken up by R cells, the R cells would transform into being virulent and would therefore kill a mouse. Ah, amazing. Thank you, Avery. Because it was from this experiment that finally the biological sciences community understood and finally accepted that DNA was the information-containing molecule of all life. And that set the stage for the genomic revolution that we're still in the middle of now. So that's the Avery experiment. If there still are any questions about that, do let me know. But certainly, hopefully, that's a little bit more clear now. Okay, so moving right along to chromatin modifications. It seemed like most of you were clear in the class about the overall structure of chromatin. That certainly didn't come up. Do know that you'll need to know that in the future for sure. The 30 nanometer fiber, individual nucleosomes, histones, and all of that. So I won't review that here because, again, that seemed to be pretty clear, but you are responsible for it. So that's what we're setting the stage for here. So we just have some very simple nucleosomal DNA. I've got 4 histones represented in purple. There are another 4 behind them giving us the octamer, and DNA is in blue. And, of course, these histones are the H2A, H2B, H3, and H4. And if we were in the 30 nanometer fiber, where we always are, we would also have histone 1 there at the exit point of the DNA, kinking the DNA, promoting 30 nanometer fiber formation, yada, yada, yada. I can't help myself. However, projecting out of what's called this nucleosome core are the amino terminal ends of the histone proteins. In other words, the beginning. Proteins go from amino end to carboxy end. This is the beginning of a protein. This is the end of a protein. It corresponds to the 5' and 3' DNA, ends of the DNA that encodes it. So the amino terminal end of a protein is its very beginning. That would start with a methionine, if you remember from genetics. And the carboxy terminal end of a protein is its end. It corresponds with the 3' end of the gene. So when we say amino terminal end of the histone proteins, we really mean the beginning of the string of amino acid yarn that makes that protein. In any event, these amino terminal tails are very rich in lysine amino acids. And lysine is symbolized with a K. Why not use an L, right? Wouldn't it be better if lysine was symbolized with an L? Well, L is for leucine, so since L was taken, we went to K. And what makes that helpful is that lysine carries on it a native positive charge. Lysine is a basic amino acid. It has a positive charge on its side chain. So why is that good? Why did I say luckily, or favorably, whatever the hell I just said? Well, because hopefully you remember that DNA is inherently negatively charged. The phosphate sugar backbone of DNA is a negatively charged backbone. And so by being positive, these lysines of these histones help the nucleosome remain stable and intact. A favorable charge interaction between the positively charged lysines and the negatively charged DNA stabilizes that nucleosome and keeps that DNA nice and tightly wrapped around the nucleosome, which is good most of the time. Because there are times when we actually would like to have access to the DNA that's wrapped in this nucleosome. And in order to access that DNA, we actually need the nucleosome to be gone. We need the DNA to be what's called naked. We need the histone proteins to be evicted. Now, that positively charged lysine that we were happy about a moment ago, we're not too happy about it now, because right now we got a nice, big, stable nucleosome where we want instability. We want to access this DNA. So how could we destabilize this nucleosome? Well, the way that we can destabilize the nucleosome is, well, perhaps, hopefully, we could get rid of these positive charges. It's the positive charges of the lysines that are making the nucleosome stable. It's the positively charged lysines that are interacting favorably with the negative DNA. So if we can get rid of those positive charges, we can weaken or loosen this nucleosome and evict those histone proteins and get to that DNA that we really need to access. So that's really what this story revolves around. We have, chemically speaking, chemical groups called acetyl groups. These are small chemical groups that can be added or removed to a variety of different proteins. Well, acetyl groups can be added to lysine residues. And when they are added to lysine residues, or lysine amino acids, those acetyl groups remove or neutralize the positive charge of the lysines. So that's exactly what we were looking for, isn't it? That's what we needed. This sounds like a good system. If we can acetylate these lysines, we could remove those positive charges, we could weaken the histone's interaction with the DNA, and we can evict those histones and get access to this DNA. So that's our key. We can acetylate these histones, remove those positive charges, and get those histones out of the way. So it sounds to me like we want to transfer these acetyl groups to our histones, right? If we could transfer acetyl groups to our histones, we can neutralize the positive charge and weaken the histone. We can get to the DNA. So what would be a good name for enzymes that transfer acetyl groups onto histones? I'm not feeling too creative, so let's just say, well, histone acetyl transferase. That seems to work, right? A histone acetyl transferase should transfer acetyl groups to histones, and that's what we want to do. And that's what we do, and that's what we call those enzymes. So HATs are histone acetyl transferases. They transfer acetyl groups onto histones to neutralize those positive charges, to weaken nucleosomes, and to provide access to the DNA. So that's a HAT. HATs loosen or weaken nucleosomes. They are activating. In other words, they grant access to DNA. They allow DNA to be accessed. They activate DNA. They activate transcription. They activate repair. They allow replication. They activate cellular processes that require access to DNA. Sometimes we don't want a loose or evicted nucleosome. Sometimes it's in our best interest to lock a nucleosome in place. Sometimes the DNA that the histones are wrapping is DNA we want to make the cell blind to. We can think of this as a liver metabolic enzyme, a gene for a liver metabolic enzyme in a skin cell. A skin cell is not in the liver. A skin cell is not a liver cell. A skin cell shouldn't be making liver metabolic enzymes. A skin cell should have an enzyme, a gene for an enzyme like that, repressed or inactivated. So we can also use nucleosomes strategically by intentionally locking DNA that we want the cell to be blind to in a nucleosome. Now, if we intentionally want to stabilize a nucleosome, that means we intentionally want all of those lysines to be positively charged. We want those lysines to have that favorable interaction with DNA so that they're very, very difficult to move, because we want the DNA that's wrapped in that nucleosome to be locked. In other words, what we really want in this case is to deacetylate those histones. So what I would like here is a complex of proteins that deacetylates histones. In other words, if there are any acetyl groups lying around that are neutralizing those lysines, I want them out of here because I want those lysines to be positive. I want this nucleosome to be stable. I want that DNA to be locked in that nucleosome. I want that DNA to be inaccessible. So what might be a good name for a complex that deacetylates histones? Scratching my head again. Again, I'm really just not a creative person, so I guess I'll call that a histone deacetylation. Can't even spell it. Histone deacetylation complex. That's a complex that deacetylates histones. That's what we want to do. And we'll abbreviate that HDACs. And that's what HDACs are. So HDACs oppose HATs. If HATs loosen nucleosomes, HDACs tighten nucleosomes. And by tightening nucleosomes, they inactivate DNA. HDACs block transcription. HDACs block replication. HDACs block DNA repair. HDACs make DNA inaccessible because they lock nucleosomes in place. HDACs are repressive. Now, what about this SWE-SNF example, or more specifically, a chromatin remodeler? What does a chromatin remodeler do? Well, a chromatin remodeler remodels chromatin. what chromatin remodelers do is they physically push nucleosomes out of the way to intentionally and actively expose the DNA that was wrapped in a nucleosome. So let's call in a chromatin remodeler to this story here. Let's say we want the Sui-SNF complex to come in and move this central nucleosome. So it comes in, grabs some ATP for energy, and it pushes, and pushes, and pushes again. Nothing's really happening. Why? Because this nucleosome is locked in place. Look at these lysines. They're positively charged. These lysines are not allowing the nucleosome to be shifted because there's such a strong electrostatic interaction between the positive histones and the negative DNA. So what do we first need to shift and move this nucleosome? We need a hat. In yeast cells, the primary hat, the one that we talk about in the lecture, happens to be the SAGA complex. SAGA is just a abbreviation for the different proteins that are in that complex. The main protein, the actual hat, is a protein called GCN5, which is what the G stands for. You don't need to know any of that. But what you do need to know is that when SAGA is called in, because it's a hat, it is going to remove all those positive charges and substitute them with acetyl groups. It is a histone acetyltransferase. Now this nucleosome is weak. Now the DNA is loosely associated with the nucleosome. Now a chromatin remodeler can take some ATP and use that ATP to do some work and physically unwind the DNA from that nucleosome and rewind adjacent DNA onto that same histone octamer and shift it out of the way. And now the DNA that was previously wrapped in that nucleosome is exposed and accessible and naked for whatever cellular process was needed. Perhaps once we've shifted this nucleosome, maybe we want long-term access to this DNA. Maybe it's time to call in an HDAC. What do you think? Because if we want this nucleosome to stay in this position for a long period of time, we better lock it down. And if we want to lock it down, we better strip away all those acetyl groups that SAGA put on and let the lysine tails go back to their native positive charge. That's going to lock that nucleosome in place over there. So it's this beautiful choreographed interplay between hats and HDACs and chromatin remodelers to loosen and shift and tighten and lock nucleosomes, constantly providing access or depriving access to specific DNA sequences that the cell wants to activate or deactivate. Yeah, that's pretty much the story. A fascinating story, I think. I spent quite a lot of time working on this stuff during my doctoral work, so it's near and dear to my heart, but it never gets old for me. Hopefully that's a little bit clearer as well, and if not, certainly let me know in the question at the end of this video. So yeah, I think that's all I wanted to say about HDACs and hats, which leaves us finally to the histone code. So what is the histone code? Well, let's blow up one of those nucleosomes that we were just talking about a moment ago. And there it is. And let's even talk about histone 1, H1. And we've already talked about these N-terminal tails, so I've just drawn them again a little bit bigger. We already said that we have the 4 different canonical histone proteins, H2A, H2B, H3, and H4. H1 kind of sitting out here, kinking that DNA. Let's even capture that, since we've got the room and the space too. And we talked about there being lysines on these tails that can be modified, and they can be modified. They not only can be acetylated, they could also be methylated. They got methyl groups added to them. That doesn't affect their charge, but it does impact the histone code. We also have serines in these tails. We have other amino acids. Prolines are here. They sometimes kink the tail, make it take a different shape. We've got threonine. Serines and threonines can be phosphorylated. So we have a whole bunch of different amino acids in these tails. And those amino acids can be modified post-translationally modified in a chemical way. They can be acetylated. We already talked about that. The lysines can be acetylated. But I already told you, lysines can also be methylated. And it's an either or. You can't be both. And acetylation impacts the charge, and methylation doesn't. Serine and threonine can be phosphorylated. And there are even other amino acids in here that can have other things done to them, such as ubiquitinylation, or like cellulic acid, cellulination. You can have a lot of different chemical groups added to these amino acids of these tails. And it's not an all or none. In other words, it's not that all of these are done, all of these post-translational modifications are done, or they're not. It's a select few are done under different circumstances, and those circumstances impact what information is encoded in these modifications. And we're not talking about the underlying DNA, right? Of course, there are A's and T's and G's. We got a start codon. There's DNA encoded. There's information encoded in the DNA sequence itself. That's the information for regulating and making proteins. We're talking about a meta level of information encoding in the post-translational modifications of these histone tails. Let me show you a few images to kind of get at what I'm saying. So what we see here on the screen is a stylized version of a nucleosome. That's what we see here. The DNA is in red, and these black lines are the histone tails. We see the histone H3 tail, the H4 tail, and then shortened versions of H2A and H2B. So same thing as what I'm trying to draw here, just a much prettier version of it, I have to admit. And we see that there are these potential modifications. We have methylations here, phosphorylations are here, acetylations are shown, and here's some ubiquitinations. And even the methyl groups, you can have one methyl group added, two methyl groups added, three methyl groups added, again, further and further information. And here what we see is that a great deal of these are done, these post-translational modifications are represented, but you need not have all of them. For example, you could have this trimethylation event here, this phosphorylation, and this dimethylation, and that might be it on this H3 tail. Maybe you have this acetylation group, and this methyl group on H4, and that's it. And maybe you don't have any ubiquitinylation on H2A at all. That particular pattern that I just described sends a signal to the cell. It tells the cell something about the DNA that's wrapped in that nucleosome. Maybe it tells the cell, silence this DNA, wrap this DNA in heterochromatin, make this DNA as tightly wound and as inaccessible as possible. Maybe that's the message it sends. Maybe it says, well, the DNA that's under you right now is primed for replication. Be ready to move, nucleosome, because you're at an origin of replication, and we're going to start replication here in just a moment. Maybe that's the information that a particular set of modifications sends. Maybe the cell has become damaged. There's DNA damage that's sensed. And in response to that DNA damage, we lay down a new histone code. For example, maybe H2B has its 120th lysine ubiquitinated and its 14th serine phosphorylated. In pink here, we see the enzymes that are responsible for those modifications. And maybe H1 is phosphorylated as well. Maybe H3 is phosphorylated at its 10th serine, its 28th serine, and its 31st serine. It's acetylated at its 9th lysine and its 56th lysine, and maybe it's methylated at its 79th lysine. Maybe all of those modifications are done as well, all in response to this DNA damage, all catalyzed by these enzymes. And maybe H4 is phosphorylated at its 1st serine, acetylated at its 5th, 8th, 12th, and 16th lysine, and methylated at its 20th lysine. This combination of modifications, that combination of all the potential modifications that are possible under this particular DNA damage, these are the only modifications that were done, and this set of modifications is a code. And it tells the cell, hey, stall in the cell cycle. Don't go any further in cellular replication, in cellular reproduction. Stop. We've got DNA damage. We've got DNA damage we need to fix. Do not progress along with the cell cycle. That's what this particular histone code tells the cell. What a beautiful system, isn't it? So there's DNA damage. The cell senses that DNA damage, says, oh my God, I can't continue on with cellular replication because we've got DNA damage. We need to fix this first. Let me lay down all of these specific post-translational modifications, and that's going to send the signal. That's going to contain the information. Pause. We have work to do. Fix the DNA that's here. This is where the damage is. It's a lot more complicated than what we're talking about, which is sometimes the stumbling block for understanding this information. It sometimes seems in all these kind of simple examples, well, how does that contain information? Like, how much information can you contain? Well, keep in mind that we're trying to keep it simple here. This is a small example of the different writers and erasers of the histone code. In orange, you have known enzymes from yeast. In blue, from flies, fruit flies. And in black, you have human. This is a small subset of enzymes that can put down markers to create a histone code and enzymes that can remove them to erase a histone code. So these are just enzymes that put down signals on the amino terminal tail of H3. How many different possible modifications are there? This many. It's even too small to show completely, but what you're looking at are the amino terminal tails of H2A, H2B, H3, H4, and H1, the linker histone. You see the individual amino acids are listed here. You might be able to make out that that's a K for lysine, we have an R for arginine, here's an S for serine. Each one of these highlighted amino acids is modifiable. It's capable of undergoing a post-translational modification. And then what you can barely see up here are all of the different modifications that are possible. I can make out here an AC for acetylation. Not sure what that stands for. This looks like a UB to me for ubiquitination. This is a SA for that adding of a cellulic acid. All of these different distinct post-translational modifications can be done to this one lysine. This tyrosine can be phosphorylated. This serine can be phosphorylated. Each one of these amino acids with black letters above it can be post-translationally modified, and each set of black letters is a different modification that can be done. Can you imagine the richness of the information that can be encoded in a system like this? If you were to phosphorylate this serine, methylate this arginine, phosphorylate this tyrosine, and acetylate this lysine on H2A, that can tell the cell something. Replicate this DNA, suppress this DNA, transcribe this DNA, silence this DNA, make this heterochromatin, make this euchromatin, repair this DNA. That can contain information, that particular code. But maybe if we just acetylate this lysine, and methylate this arginine, that sends a different signal to the cell. No, don't replicate this DNA, repair this DNA. Don't suppress this DNA, activate this DNA. Don't transcribe this DNA, repress this DNA. Who knows? But it's another code. So by choosing and modifying which post-translational modifications are added to which tails, you can embed and layer information on top of the DNA. This is epigenetics. This is an epigenetic mechanism. The histone code is an epigenetic phenomenon. You might have DNA that lies within this nucleosome that says, be tall, pea plant. Be proud, pea plant. You've got the dominant gene to be tall. That might be what this DNA codes for. But if the histone code of the nucleosome wrapping that DNA says suppress and silence, suppress and silence, suppress and silence, that gene will never be expressed. That phenotype will never be observed, and you will have stood on top of the genetics. That's what epigenetics is. And by standing on top of the genetics, you would suppress a dominant phenotype and see a short pea plant. Our environment, our experience, our choices all impact this epigenetic type of imprinting. And these are inheritable. We can pass these modifications onto our children and onto our grandchildren, in fact. Some of these epigenetic changes are so long-lived they persist 2 or more generations. So we alter our DNA with our very existence. Our life experience changes our DNA, not at the nucleotide level, but at the epigenetic imprinting that stands on top of it. And by making DNA masked and silenced through the histone code, we can make it as though those genes aren't present in the cell at all. It's phenomenal. It's incredible. It flies in the face of all that we tend to think about our DNA. It's very Lamarckian rather than Darwinistic in the sense that our environment and our experience can alter our appearance, but it's very much true, and we now understand the molecular mechanism for it. So that's the histone code, and hopefully that as well makes a little bit more sense and is a little bit clearer. So that's it for this review. 40 minutes, not too bad, I guess. Hopefully some interesting stuff, hopefully some clarification. If I did not cover the information that you had wanted covered, please let me know and get back to me so that we can meet one-on-one and do a little bit of more intensive review for you. Also, please be aware, if you haven't seen it already, that I am leaving robust feedback to each and every one of you in Edpuzzle. So whenever you express confusion, I'm trying to clarify that for you in Edpuzzle. So please go back to those Edpuzzle videos after you see that they've been graded and click on the comments link to see what I've said in reply to you. Many times there's a lot of rich information that I provide to you specifically in Edpuzzle, and I find out oftentimes near the end of the semester that students didn't even know it was there, and it's almost like a horribly lost resource. So be going back to Edpuzzle, even in addition to watching the videos like these, so that you can see the specific and personalized feedback that I'm giving to you about the questions you conveyed to me. Alright, I guess that's it. Thanks for watching, and if there's anything you'd like to tell me, anything you'd like to pass along to me, anything you continue to think I should know about your level of confidence with this material, please let me know right now.